Welcome to Grow Bio channel. In today's video, we're gonna see about gene regulation in prokaryotes, Trp Operon. If you want to know more about gene regulation and other operons such as lac operon, I will give link in description below. Now we can see about tryptophan operon. The tryptophan operon is the repressible operon for the anabolic process. That is, it is the anabolic process. The synthesis of tryptophan is made by the Trp Operon. Why it is called as repressible operon? Okay, because the tryptophan, the byproduct of Trp Operon, tryptophan itself acts as the inhibitor or repressor for the own reaction. So it is known as repressible operon for anabolic process. The tryptophan is one of the essential amino acid. It is produced constantly by the bacteria and stops when available at the cytoplasm, that is after producing significant amount, or when available in the environment. That is, the tryptophan is the essential amino acid for whom? For humans. Okay, it will be produced by the bacteria for its own concern till the significant amount is achieved. It will be needed by the bacteria itself for synthesizing or for propagating itself. Okay, or when available in environment, it means when the bacteria is cultured in the artificial conditions such as laboratory, there the media itself contains the tryptophan. When there is tryptophan in the media, the bacteria won't produce it because it is a wastage of energy. So this is called as repressible operon. Okay, in this the tryptophan itself acts as the repressor, as the example of feedback inhibition. Okay, so it is known as repressible operon for the anabolic process. Next, now we can see about structure of Trp Operon. First there is Trp R. Okay, it is the repressor, and then after some gene, okay, there is a break. After some gene only the promoter operator sequence is there. Okay, the promoter is the full gene. Only a certain part, the last half will be operator. Okay, this is a combined gene, promoter operator. And next is Trp L, that is Trp leader sequence. We can see about it later. And next is structural gene, EDCBA, which is reverse of ABCDE. In D, you can see another promoter sequence. So when tryptophan is absent or low, the tryptophan must be produced. So the holorepressor. What is holorepressor? It is combination of co-repressor and aporepressor. Okay, the active form of this complex is known as holorepressor. It must come out of the operator. Okay, then only what happens? The transcription will occur. So what is co-repressor? It is the tryptophan. When tryptophan is low, then the co-repressor will be not there. So aporepressor will be in inactive condition. So they will move away from the operator. Okay, it unwinds from the operator and makes the complex inactive. So the transcription will occur and more tryptophan will be produced. Now, why the tryptophan synthesizing enzyme is constantly produced? Why the structural gene is always on? So answer is, it is used by the bacteria for its growth and propagation, and it is also used by the host system. It is the base for many hormones. Okay, for example, serotonin. The serotonin is a neurotransmitter. It is responsible for well-being and happiness of the human body, and other processes such as learning, memory, and other physiological processes such as vasoconstriction, vomit taking, etc. So that is nausea, etc. So these conditions will be depending on the serotonin, which has the base as tryptophan. And next is melatonin. It is responsible for sleep-wake cycle. It is very important for maintaining the homeostasis in the body. And next is vitamin B3. It is used to treat disease like pellagra. For these three main neurotransmitter, hormones and vitamins, the base is tryptophan. So it must be needed by the body. Now we can see about Trp Operon structural genes. Okay, what are all the enzymes produced by the structural gene? First is Trp E. It produces anthranilate synthetase. It is called as epsilon. Okay, the enzyme produced by Trp E is anthranilate synthetase. And Trp D produces anthranilate isomerase, that is delta. And Trp C produces INGP, that is indole glycerol phosphate synthetase. Okay, it is an enzyme. And trip B produces tryptophan synthetase beta, and trip A produces tryptophan synthetase alpha. Okay, now we can see the biosynthesis of tryptophan. When presence of core is made, 2 epsilon and 2 delta, that is anthranilate synthetase and anthranilate isomerase, will produce anthranilate. Okay, the same enzyme will be responsible for producing phosphoribosyl anthranilate. And when INGP synthetase is acting on phosphoribosyl anthranilate, it produces carboxyphenylamino deoxyribulose phosphate. Okay, again in presence of same enzyme INGP synthetase, it produces INGP indole glycerol phosphate, and in presence of 2 beta and 2 alpha, that is tryptophan synthetase gene product, it produces tryptophan. This is the biosynthesis of tryptophan. It is easy 5-step process. Okay. Next we can see elaboration about Trp Operon. Trp Operon consists of Trp R, and you can see a break there. Okay, which means there is more gene in between that. Okay, afterwards only the promoter operator sequence will be there, and Trp L will be there, and then structural gene EDCBA will be there. The Trp R, PO and Trp L, these are upregulatory element, and EDCBA is known as structural element. What is upregulatory element? It is the regulatory element which maintains the gene switch on and switch off for tryptophan synthesis. Now we can see condition 1. When tryptophan is present, what happens? So when tryptophan is present, the Trp R produces many repressor protein, but it is inactive protein. Okay, it can't go and sit in the operator region. It needs another help from co-repressor. The co-repressor is nothing but tryptophan itself. As we already saw, tryptophan itself acts as the repressor. So it acts as the repressor. So the holoenzyme will be bound to the operator by forming aporepressor co-repressor complex, which is also called as holorepressor. Okay, the repressor gene product aporepressor, in presence of co-repressor, that is tryptophan, it produces holorepressor. Okay, which is the active form of repressor. So what happens when it sits in the operator? Then sigma factor can't be recognized by the promoter. So what happens? The transcription will not occur. Okay, when the holorepressor forms, answer, the holorepressor forms only when significant amount of tryptophan produced or when the tryptophan available at the environment. Then only the tryptophan will be more, and then it acts as the co-repressor for its own synthesis. Okay, then only the holorepressor will be formed. In condition 2, when tryptophan is low or absent, what happens? So here you can see the bounded co-repressor and aporepressor at the operator region. When the tryptophan is absent or low, the tryptophan must be produced. So the holorepressor must come out of the operator. When tryptophan content is very low, then the co-repressor will unwind the repressor, aporepressor. Because co-repressor is nothing but the tryptophan itself. When tryptophan content is low, what happens? The co-repressor will unwind the aporepressor and makes the aporepressor inactive. Okay, it is the inactive form of protein, so it becomes inactive. Then what happens? The transcription occurs and more tryptophan will be produced. The scientist mutated bacteria and eliminated the Trp R. Then what happened? So why they mutated? Because humans are in need of more tryptophan. So what they did? They mutated the bacteria for Trp R. When Trp R is not there, what will happen? They thought like more tryptophan will be produced by the bacteria. But what happened? It doesn't happen like that. Okay, there is only limited production of tryptophan, not more, because there is other regulatory gene present in the Trp Operon to regulate the tryptophan production. It is called as Trp L, that is Trp leader sequence. Okay, now you can see Trp Operon structure. There the trip L is elaborated there, okay, in below structure. First is red color, that is region. In that region, the base pair 10 and 11 code for tryptophan. Okay, so there will be 2 tryptophan will be produced by the red region. Okay, next gene, in that itself there is green region. In 60 to 68 base pair, it is the region 1. And brown region 75 to 83 base pair, that is known as region 2. And 110 to 121 base pair is known as region 3. And 126 to 134 region is known as region 4. In this gene itself there are 4 region, and other region which is responsible for tryptophan synthesis. The 10th and 11th base pair will be responsible for tryptophan synthesis. So now we can see about trip L, that is trip leader sequence. In prokaryotes, there will be simultaneous transcription and translation because there will be no nuclear membrane will be present. Okay, so there will be a transcription and translation at the same time. The trip L sequence forms stem loops in the region because of the complement base pairing. Okay, there are 4 region, it forms 3 stem loops. Now we can see about that. The region of 10 and 11, that is at 10th and 11th base pair, it contains 2 trip codon, that is UGG. In 64 codon, only 1 codon will code for tryptophan, that is UGG. Okay, so the stem loop base pairing can occur in between region 1 and 2. It gives pause for the tryptophan synthesis. And region 2 and 3 will code for anti-terminator, that is the sequence, that is the transcription will not end when region 2 and 3 stem loop forms. And then region 3 and 4 stem loop, it is transcription terminator, that is the Trp Operon will stop the tryptophan biosynthesis. Okay, so 4 parts of region forms 3 stem loop structure. We can study about it in detail. Okay, now we know about trip R and trip L gene and what it does. Now we can see about one condition. When tryptophan is low or absent in the cell, what happens? There will be simultaneous transcription and translation that will occur in prokaryotes. There will be no tryptophan tRNA, that is tRNA will not have tryptophan. What is the work of tRNA? The tRNA will bring the amino acid sequence to the ribosome, okay, so that the polypeptide chain will be formed. When the tryptophan itself is not present, there will be no tRNA to contain the tryptophan. So there will be lack of tRNA to join the ribosome to produce the protein. So the ribosome waits for tRNA. So what happens? The ribosome will become late, that is translation will become late. So the region 2-3 stem loop structure will be formed. That is the signal for anti-terminator, that is the tryptophan synthesis will continuously happen when anti-terminator signal is produced. Then afterwards what happens? The mRNA will bind to region 3. Okay, so when ribosome translates the protein, the RNA polymerase, what happens? The transcription is also happening there. Okay, now due to complementary base pairing, region 2 and 3 loop will be formed. In region 3, which has more GC content, so the bonds will be very strong and the expression proceeds to structural gene. So the tryptophan synthesis of EDCBA will be translated and then tryptophan will be produced. Okay, here this is more important because in region 3 it has GC content more, so the bond will be strong, so the expression proceeds. Okay, now we can see another condition. When tryptophan is in significant amount, this is intracellular. Okay, consider it is produced by the bacteria itself. Okay, when tryptophan is more, that is significant amount is produced, then what happens? The tRNA will be more because the tRNA will have tryptophan in it. So no pass will occur at 10th and 11th region. Okay, so the ribosome will cover region 1 and 2 very fast and tRNA binds to it. and translation occurs simultaneously. So what happens? Because of no pass, the 2-3 loop cannot be formed, okay, but the 3-4 loop will be formed. Because 2-3 loop, why they didn't form? Because ribosome will be present at the 2 region, so 2-3 loop will not be formed, but 3-4 loop will be formed because of complementary base pairing. What does 3-4 loop indicates? It is the terminator loop. There will be more adenine guanine, there will be more adenine thymine content, which is the weak bond, because it has only 2 hydrogen bond comparative to GC content which has 3 hydrogen bond. So AT is weak bond. So what happens? The bond breaks, and what happens? The ribosome small and large subunit will be fall off, and promoters based RNA polymerase also falls off. So what happens? The structural gene expression doesn't occur because of breakage of AT bond. So no transcription will occur. So no tryptophan biosynthesis will be occurred. Okay. So when tryptophan amount is achieved, then tryptophan biosynthesis will not occur because of weak bond of AT in the region 3. And when tryptophan is low, then what happens? The pass occurs and region 2-3 stem loop will be formed because of complementary base pairing, and region 3 will have more GC content, and what happens? The expression proceeds and tryptophan synthesis is occurring. Okay, this is the 2 condition. And condition 3, when tryptophan is present in significant amount in extracellular, that is in lab condition, when the tryptophan is present in the media, what happens? There will be enough tRNA present for translation. So ribosome will bind to 2-trip codon of TRPL. There will be no pause. Okay, so the reaction will occur very faster and terminator stem loop will be formed. Okay, so 2-3 stem loop will not be formed here, only 3-4 stem loop will be formed. So then in TRP-R, the co-repressor will bind to aporepressor because of presence of tryptophan. The tryptophan acts as the co-repressor. Okay, the co-repressor will bind to aporepressor. So transcription will not occur. Okay, when significant amount of tryptophan is present in extracellular, or when tryptophan is produced more at significant amount, then what happens? The tryptophan biosynthesis will not occur. Okay, when tryptophan is not present or absent, what happens? The anti-terminator signal 2-3 loop will be formed, and then the expression proceeds to form the tryptophan. Okay, hope you understood. If you have any doubt, let me know by commenting in the comment section below. Subscribe and please press the bell button below. Thank you.